Hi, so good afternoon. I'm pleased to discuss issues pertaining to the science and regulation of food from genetically engineered animals. I'm one of three co-authors of the paper that we'll discuss this afternoon. Allison Van Eenenum was the chair of the committee. She's from the Department of Animal Science at the University of California at Davis, and Bill Muir is in the Department of Animal Sciences at Purdue University. This was a peer-reviewed paper. We benefited from the comments of David Edwards at the Biotechnology Industrial Organization, of Greg Jaffe at Center for Science in the Public Interest, Paul Olin at California Sea Grant, and Mark Walton at M. Walton Enterprises. Our cast liaison was David Scarf from the American Veterinary Medical Association. By way of introduction, genetic engineering is the use of recombinant DNA techniques to insert genetic material from one organism into another. The upper right, I show what we call an expression vector. It contains the growth hormone gene from common carp in this example, with the appropriate regulatory elements to guide its expression in the host. Below, I show the micro-ejection of this construct into the recently fertilized eggs of walleye, a freshwater fish. The hope here would be that some small percentage of the fish that were so micro-ejected would take up the gene, would express it, and would transmit it to their offspring and have a favorably altered phenotype. Genetic engineering would be used to introduce new traits into an organism or to produce a useful biological substance such as a therapeutic protein. Genetically engineered organisms may also be referred to as genetically modified, transgenic, or bioengineered animals, or as living modified organisms. The goals of genetic engineering are several. They could be used to enhance production attributes, for instance, to produce a fish that grew faster than the non-modified counterpart, or to enhance food quality traits, for example, to produce more lean muscle mass, as we see in the transgenic pigs at the lower left. Gene transfer might be used to improve animal health, for instance, by improving disease resistance in the host, to produce products intended for human therapeutic use, for example, pharmaceutical products, to enrich and enhance the animal's interactions with humans, for example, new color varieties of pet zebrafish or glowfish that I show at the lower right, to develop animal models for research purposes, for example, pigs as models for cardiovascular diseases, or to produce industrial or consumer products such as fibers for multiple uses, such as spider silk in the milk of transgenic goats. Many different animals have been genetically engineered, although relatively few have moved from the laboratory to commercialization. Transgenic animals such as mice, rats, and zebrafish have become increasingly important for biological and biomedical research. In 2009, the first genetically engineered animal producing a pharmaceutical product, a goat synthesizing a recombinant human antithrombin 3, an anticoagulant in its milk, was approved by FDA. However, to date, no genetically engineered animal intended for use as food by humans has received regulatory approval. This last statement raises the issue of how genetically engineered animals are currently regulated. Some of the controversy regarding such animals stems from issues of regulatory oversight. Now, regulatory authority over the products of biotechnology was not established by an act of Congress. Rather, in the 1980s, the White House Office of Science and Technology Policy promulgated the Coordinated Framework for the Regulation of Biotechnology, which extended the scope of existing laws to establish oversight of production, field testing, and marketing of genetically engineered organisms. Agencies with authority under those laws then began rulemaking to implement the Coordinated Framework, focusing at first on genetically engineered plants and microbes. Genetically engineered animals were not a subject of discussion until later. The regulatory approach taken by the Coordinated Framework proved controversial, as did aspects of its implementation. Under the Coordinated Framework for genetically engineered animals, the scope of the Federal Food, Drug, and Cosmetics Act was extended, leading to oversight by FDA. The FDA considers the recombinant DNA construct to be a new animal drug because it is an article intended to alter the structure or function of the animal. New animal drugs must be licensed based on a showing that the product is safe and effective for the intended use. In 2009, FDA made public a framework regarding how genetically engineered animals would be regulated. FDA's guidance for Industry 187 laid out a 7-step process for oversight. Molecular characterization of the recombinant DNA construct focuses on whether it contains DNA from viruses, bacteria, or other organisms that pose public health risks. Molecular characterization of the genetically engineered animal lineage assesses whether the new gene construct is stably inherited and expressed in the transgenic line. Phenotypic characterization of the transgenic line focuses on whether the line looks like, reaches developmental milestones, and proves disease-resistant like appropriate comparison animals. The durability assessment asks whether future generations of the genetically engineered line prove equivalent to the genetically engineered animals evaluated during the approval process. The food safety assessment asks whether food products for the genetically engineered animal have the same composition as non-engineered animals. And if there are differences, is there reasonable certainty of no harm to the consumer? Key endpoints include allergenicity. In the environmental assessment, FDA must meet the requirements of the National Environmental Policy Act. It must reach either a finding of no significant impact or develop a full-blown environmental impact statement. These first 6 steps comprise the safety assessment. The effectiveness assessment is addressed in item 7. The claims assessment determines whether the genetically engineered animal performs as expected. Does it grow faster? Does it produce the desired pharmaceutical protein? Only with successful passage through all 7 steps would the FDA license commercial production of a genetically engineered animal. We should note that approval can be limited or revoked should adverse outcomes be observed. We should note that the FDA has chosen to exercise enforcement discretion with regard to certain classes of genetically engineered animals, such as those of non-food species that are raised and used and contained in controlled conditions. For example, FDA exercised discretion and allowed Yorktown Technologies to market genetically engineered zebrafish as aquarium pets called glowfish. There's no intent to enforce the new animal drug requirements on genetically engineered animals used in research institutions. For example, genetically engineered mice that are commonly used in biomedical research. The FDA has reserved discretion to take enforcement action later if safety concerns associated with these genetically engineered animals are identified. As I indicated, the current regulatory process has its proponents and detractors. Regarding strengths of the oversight regime, the FDA pre-market review of product safety and efficacy is widely regarded as rigorous, and it is mandatory. Approval for marketing can be contingent on adhering to specified methods of containment and production known as limitations for use. These limitations may have important bearing, for example, on the environmental safety of producing genetically engineered animals. Lastly, agency approval of marketing of a genetically engineered animal is followed by monitoring, and approval for a product to be withdrawn if adverse outcomes are subsequently observed, although critics note that the latter regulatory process can be long. Among weaknesses over the oversight regime, the regulatory process is not necessarily publicly transparent. By law, the FDA cannot even disclose what products are under regulatory review. While the FDA has legal authority to consider animal health and safety, it lacks authority to consider social concerns, including animal welfare considerations that are important to many in the American public. Some detractors note that data required for regulatory review are produced by the corporation seeking approval for its product, not by the FDA or by an independent body. Others counter the provision of these data appropriately is the responsibility of the proponent of the genetically engineered animal. Lastly, there are no provisions that deal specifically with how to handle environmental risk. That is, NEPA is procedural. It tells FDA how to evaluate risk, but not whether and how to go forward in the face of non-zero risk. Some detractors question the expertise of FDA in environmental risk assessment. Another set of criticisms of the current regulatory process regard the labeling of genetically modified foods. By law, the FDA cannot require the labels include information about production methods if there's no material difference in the product solely due to the production process. Further, in a landmark 2002 study, the National Research Council found that foods from genetically engineered animals did not present different or greater safety concerns than their conventional counterparts. In cases considered to date, the FDA found that foods from genetically engineered animals did not differ materially in nutritional value, organoleptic properties, or functional characteristics. Hence, FDA does not consider the fact that a food was made using genetic engineering to itself be a material difference. On the other hand, voluntary labeling of food is allowed if the label is neither false nor misleading. Hence, food may be labeled as containing GM ingredients or as GM-free, so long as that information is